This study was for patients with resected or early stage HER2-positive breast cancer tumors. So specifically, we wanted to test whether the standard of care currently, which is trastuzumab, could be improved by adding a second anti-HER2 therapy such as Lapatinib. What we did is that we conducted a prospective global trial with a lot of collaboration in which we assigned patients to receive the standard chemotherapy, surgery, and after that they could receive anti-HER2 therapy in the form of trastuzumab alone, trastuzumab followed by Lapatinib, or the concurrent or dual administration of the two anti-HER2 treatments. It came out that Lapatinib did not add to the benefit of trastuzumab. So in terms of efficacy, we showed that trastuzumab should remain the standard of care anti-HER2 treatment for patients with HER2-positive breast cancer. We also found that this treatment was actually very safe, especially from the cardiac standpoint, as fewer than 1% of patients had any significant cardiac toxicity. This is very important for clinical practice. The efficacy was much better than we had anticipated. More than 95% of the patients are alive after 4.5 years of median follow-up, and approximately 86% of the patients are alive without breast cancer. So it reflects the improvements we're making on the field overall. It also lets us know that we need to do still some translational work to correlate some of the molecular findings in the tumors, to identify the subgroup of patients who do so well with trastuzumab, they don't need anything else, and other patients who may benefit from participation in new clinical trials. The clinical implications include that trastuzumab added to chemotherapy remains the standard of care globally for patients with HER2-positive breast cancer. Also, that this therapy is very safe from the cardiac standpoint. And the last but very important finding from ALTTO for practice is that in ALTTO we could not corroborate that the dual anti-HER2 blockade, which had been predicted to lead to a better survival based on the results of the neoadjuvant study called NeoALTTO, could not be corroborated. So ALTTO really calls into question the concept that we could use the results of neoadjuvant studies to predict what happens in the most important setting, which is the adjuvant setting. This is a very important point because many people, including ourselves, we felt that the early surrogate of pathological complete response findings could explain or predict what would happen in the adjuvant setting, but unfortunately that is not the case. And now this has been shown in patients with hormone receptor-positive breast cancer. It has been shown with bevacizumab in patients with triple-negative breast cancer, where bevacizumab improves pathological complete response in triple-negative, but it does not improve disease-free or overall survival in the adjuvant setting. And now we have the third subtype of breast cancer, HER2-positive breast cancer, in which we are showing the same thing. The findings in pathological CR do not appear to correlate with ultimate patient disease-free or overall survival. Bottom line, Lapatinib does not add to the benefit of trastuzumab. Trastuzumab on its own leads to much better outcome than we had anticipating, and this therapy is very safe for patients. I think in addition to these clinical results of drugs per se, is the fact that we are finding molecular markers in the tumors that are helping us predict outcome of patients. Specifically, we did a presentation in which we have identified a 14-gene profile that appears to correlate quite well to benefit to trastuzumab. And I think that is really a hint of the future. We need to better understand biology, and now that we're getting the results to reach that point. That it's important to collect tumor specimens, it's important to share tumor specimens with investigators so that we can understand their underlying basis of breast cancer to develop better treatments. There are two different areas for young women diagnosed with breast cancer. Number one, can we do something to help preserve fertility? And some data were presented here at ASCO related to this issue. And the other issue is how we should treat their cancer per se. So for some women in which physicians and the patient decides to proceed with ovarian function suppression, then one of the presentations here at ASCO showed that using an aromatase inhibitor, specifically exemestane, led to better outcome compared to tamoxifen. But again, the central question, which will be answered later this year, is whether ovarian function suppression should be part of standard of care for premenopausal women with breast cancer.